Okay, I'm using the computer audio. Yes. Let's make this big. Start the video. We already got some participants. All right, I got stuff behind me. Apparently, I gotta erase that. Okay, and everybody has logged in with their nickels, my nickels, because if you didn't do that, we're gonna have problems. So if you didn't, log out and log back in. It should come up your first and last name. So if you did not log in with my nickels, log out and log back in. We got plenty of time. I'm gonna have myself a little like checklist of things I wanted to cover with y'all. There we go. Oh, that would be awesome. Yes. Okay. Erase this stuff. Not behind me. Okay. All right. So do this. And I'm going to share my screen. Okay. So if you did not hear my announcement just a few seconds ago, you need to make sure you are logged in through My Nickels. That's your single sign-in, so that you effectively get into your groups. Because if you didn't do that, our breakout rooms are going to fail again. And we want our breakout rooms to be successful. Okay, it looks like we got first. Usually if your first and last name comes up, then you've logged in correctly. So we're still waiting on a bunch of people, but that's okay. Class hasn't really started yet. Okay, oh, I need my chat open so that if people say something. Good morning, all. Good morning. How are you? I love the positivity. And it's Friday. Happy Friday. I'm excited. Today is my son's favorite day. And I am recording, right? It's hard to see if that light is flashing with this protective gear on. And the classroom's open right now. So if you ever want to join in face to face, this is where I'll be doing the recording unless something happens. But regardless of whether I'm here or not, you can always come in this classroom for this Zoom meeting. So give everybody, we got about 3 more minutes. Anybody have a good joke they want to share this morning? You can turn your audio on if you have a really great joke you want to share. If not, I have a really cheesy one. Okay, so this is one my son learned. What is a tree's least favorite month? Not fall. It is September. But fall's a season, not a month. Oh, I got some claps. Yay. These are the only kind of jokes I know. Really, really, really cheesy ones. Oh, I have another one. This is my favorite. He hated this one, but I really like it. Okay, what do a marine biologist and a dog have in common? A dog wags his tail. A marine biologist tags a whale. Nerd jokes in the morning. You might want to grab a claw. I think it's good. Let's see. I have a plethora of them. If y'all are good with me entertaining you for the next 2 minutes. Oh, what do you call an acid with an attitude? Yes.
Sassy acid.
Amino acid. Amino acid. I like that one. I like that one. All right, so my son's, y'all heard my son's really into Legos. So I have to go out and actually find Lego jokes, which is really hard. But so what is the most common surgery in the Lego hospital? Plastic surgery. Yeah. Yeah, and this is why I don't do stand-up entertainment. I'm gonna stick to my day job. I'm glad some people are entertained. That's a good thing. We have a few people in the classroom. So anybody who is not, you don't see them on the thing, they're here in the classroom. That was the best one. Courtney, you're very sweet. Yeah, so my deal is I have to give my son a joke in his lunch every single day. He gets one taped to his lunchbox because he brings his lunch to school. So yeah. Towards the end of the year, they start getting worse and worse because I have a harder and harder time finding good ones. Oh, he saves them all. He saves them all. I went to his lunchbox actually leaked yesterday, and it got on the little paper that I wrote his joke on, and I went to throw it away, and he like flipped out on me, and he was like, no. It's okay, I'll let it dry, whatever. So yes, he's a sentimental at 8 years old. He's already sentimental. Okay, so I think we can start. We're 10:45. So everybody should have logged on through their MyNichols account on the Zoom, because you are going to need to be on the Zoom, and you are going to do some breakout room stuff. So make sure you're on the Zoom and that you went in through MyNichols. Okay, if you didn't, take a minute, log off, and get back on going through MyNichols. We're not really going to wait for you, but it's okay. It's okay. All right. So the first thing I want to do is I want to show you too many things going on over here. Okay. I want to show you, that is not what I want to show you. Okay. So let me move this. So I want to show you, this is kind of where I'm trying to put things. So what you should have watched, right, are all the pre-recorded lectures for this chapter. You should be up to date on this chapter, and I'll be posting more, maybe tonight, maybe tomorrow morning, but I'll be posting, and when I post them, I will put an announcement, and I'll let you know that they're up. But you're always going to go to this link right here, okay? So you should have that done, and
Someone just asked, when's the self-check for chapter 1?
it's not ever going to be due. I'm never going to grade you on it. I figure you are juniors and seniors, and if you want to evaluate how you're doing, more power to you. You're going to be more successful. If you choose not to, that is your own personal choice. But they're really good questions. They're worth doing. They're absolutely worth doing. So, but I would do them after our last, like today's our last day on chapter 1. You will have gotten everything, because the self-check is the entire chapter. So you want to wait until you've basically done the whole chapter before you try to do the self-check. Otherwise, there are going to be things that you don't know on the self-check. So wait till the end and then do it. Make sense? We good? I don't know why I'm giving a thumbs up. You can do the self-check. I think what I tried to do is to allow you to do the self-check as many times as possible. Like there was one, I think there was one option that said like unlimited. And that's what I checked. So it's never going to give you a grade, but you can do it as many times as you want. And it gives you feedback after you submit the quiz. Hopefully I did all that right. Yes, we are using the Reef app today. Yes, we are. Yes, we are. I'm giving everybody a chance to make sure they're on Zoom in the right way. And then we're going to do our normal attendance. So if you just give us just a second, I'm going to show you a few things on Moodle, and then we're going to use the Reef app. So we'll use it for attendance, and then we'll do some iClicker stuff. Okay, so this was yesterday's discussion. So I wanted to tell you what I'm going to do with yesterday's discussion. There are a few parts of this discussion that are not quite done yet. So a lot of them look really, really good. But see, there are some that need to be finished. So if y'all go in and finish them, what I will do this weekend is I'll look at them and then I'll add some comments, and I'll do it in like a highlighted color or something so it stands out. So that I make sure that you're putting the right information in, and I can add comments. And I mean, like, this is an amazing study tool. So I'm really hoping that this allows you to keep track of all that stuff. How do I know if I logged into Zoom using my Nichols email? You go to mynichols.edu, and that's your single sign-in. Then you go to Moodle, and then you click on our Zoom within the Moodle page. So if you're on Moodle, if I can go up, you're going to click on this link right here. So you log in through My Nichols, go to Moodle, and then click on this. Okay, is that good? Gonna assume so. Okay. My sidetrack. Where was I? Oh, I was talking about our slides here. So I know it didn't quite work exactly as we wanted last time, but we will all get better, and we will all hopefully it'll get better as the year goes on. So use it, go back and check them. And so my request to you is, you were given one to do. So if you didn't finish it, go in and finish it as a team. And then go and check the one above you and the one below you. The only difference would be if you're like at the end of the list, if you're hydrogen over here, what you would check is helium, and then you would go all the way up and you would check sulfur. Okay, so you finish your slide, check, and then I will finish your slide. So what I want to do is I want to give y'all about 5 to 10 minutes, maybe less, I don't know, to go in and check these right now. That way you've really spent the time on it before I go in and check it. Okay, so I'll give y'all a few minutes to work on them. And I'd like to try to put you into your groups. So I'm going to try and do that again, and we'll see what happens. So breakout rooms. I'm going to open all rooms. Why do I have people not assigned? I have so many people not assigned.
Whenever we did it,
Okay, y'all work on that.
they had trouble.
And
I don't think it works.
I will try to assign you to places. Are y'all all in your rooms? The ones who did? I'm hoping so. Okay, and then I'm gonna go, where's mine? Here, okay. Oh, that's not what I wanted to do. I need my sortable list. This is not sortable. That's okay. Okay, sodium. See, I don't think as I assign it that it's gonna keep. Somebody told me it was just because y'all hadn't signed in. But that can't be that that not many people are not, oh, y'all are not joined? I'm not in a room. It looks like it's saying that all of these people have not joined, that all of these people who are, can y'all see the breakout room in progress? Can y'all see this screen? Annie, do you see the screen?
It's the part 2 screen.
Yeah, oh, I could be projecting for y'all who are in the classroom. I'm sorry.
Well, I mean, I'm on Zoom, so then-
Yeah,
I'm on Zoom.
Oh,
but he's not on Zoom. Okay. So last session, but perhaps we need another invitation, not in a room. I am going to take a screenshot. Oh, this is not a Mac. I hate that this is not a Mac. What's a screenshot shortcut?
Command Shift 4.
Like Control Option. What does it say? Command Shift? No. But there are so many people. Alt Control Function Print Screen. Oh, why did I open Excel? Okay. But was not, but it was supposed to be in magnesium. See, y'all are logging in through Moodle. I don't know why y'all aren't joining. Okay, I'm locked out of my Zoom account on my Nichols email. Cameron, you weren't able to log into my Nichols? Alt Control Function. Let's just see if we do this print screen. Maybe it's because this is a smart board, it's not doing it. That's awesome. I made sure to log in. And you didn't get put in a room. Okay, so they told me that was the problem. That must not be the problem. Must be something else. Because there are too many of you who were not assigned. But that's okay. You know what groups you're in, and you know what slides you're supposed to be checking. So I will go back and I will say that did not fix our problem. And y'all work on those slides. Taylor, what group are you in? What's your element? Taylor. Fluorine. Fluorine. So we should have 3 other people in fluorine. Where do we go for the slides again? Oh, so you go to Moodle. You go to Moodle, and under chapter 1, I have all the stuff for chapter 1. And so we had a discussion part 1, and then we're going to finish the discussion part 2 today. But right here, discussion part 1 is the slides. Okay. All right. So inside of there, where was that? Here. You also have your groups. So if you've forgotten which group you're in, you can go and find your groups. Okay, so if you were in beryllium, these are your other 3 people who should be in your group and can join the breakout room. Okay, well, I'm not going to worry about breakout rooms today because it's going to take me too long to sort everybody. So we're just going to not worry. So I'm going to close all the rooms, right? And everybody can work in the common thing. See, I don't understand, but it's saying not joined, but then it's got people in the room. I don't understand. See, I don't understand. People are some people are in all by themselves. I don't know. Makes no sense. Okay, I was gonna move down here. I was just kicked out. Yes, y'all were just kicked out because the breakout rooms are not working for everyone. So we're all gonna have to suffer. And then I'm gonna go back and tell them that did not fix our problem. I know. It's something on my end. I don't know how to join the breakout rooms. That's something on my end, Ross. And I was told that it was just a login error on y'all's end, but I don't think that's true. I think there's something else going on. So I'm gonna have to work on that. That's gonna be a work in progress. So I'll still give y'all about 5 more minutes to finish up on the slides. They're looking really good. I think this is going to be a really good tool for y'all to use. And if as you're doing this, you have some suggestions on what to do, Christian Landry. Oh, that is exactly what I need. I need somebody who can successfully do it. Dr. Christian Landry. I'm writing that down right now. Thank you. Thank you, Hannah. Landry. Okay, she's in biology, pathomicrobiology. Awesome. Thank you. Christy. Christy. Okay, got it. Thank y'all. We will get it figured. We will get it figured out. Okay. So, close this. So I'll give you all about another 2 minutes. While y'all are doing that, I'm going to do the cloud. Oh no, where is that? Oh, it is there? Oh yeah, it's there. So I'm going to start our class. All right. Okay, and when you get a chance, we're gonna go ahead and check in. And you're gonna tell me, are you present at home or in the classroom? And pretty much everybody's gonna say they're at home. Okay. Multiple choice. So y'all should be finishing up the slides, and you could do that. Because then we're going to use the iClickers for real, and we got some questions to answer. It's so hard not seeing anyone's faces. No, well, yeah, you can. You can. You can turn off the Zoom. Yeah, you can turn off the Zoom. If you're in here, I'm not talking to anybody at home. We have a few people in the classroom. Yeah, you can turn off the Zoom. Yeah, absolutely. Okay. Should have a few more than 51 responses. Okay. All right, I give y'all until 1 minute, 45 seconds, and then I'm closing the attendance poll, and we're gonna do some iClicker stuff. So some review questions. Anybody else? Because I'm about to close it. Okay. And I just got, where did my chat go? My chat went. Isn't it mandatory? It isn't mandatory. No, so, no, you have to stay. You have to stay. Oh, these are are y'all people who couldn't do it on the cloud, on the iClicker? Hannah, Ross, Hannah, Christian, Jacob? It is mandatory to come to class on Fridays, but it's really a digital Friday. Yes, still can't get the app. Okay, Ross, I gotcha. You answered by clicker. Yes, I have a physical clicker linked, but it says no base.
Yeah, I put my stuff into the app.
It should work. So physical, and it says, I'm writing this down, no base from, okay. I will look into, Christian, I will look into that for you. So Friday's supposed to be all digital, but the classroom's open during our time. So I am saying you can come in if you want. We just can't have more than 32 people in the room. Okay, so let's do some iClicker. You don't have to be a certain distance from the classroom with the iCloud clicker, which was what I use. You just have to have internet. So if you're on Zoom, you should be able to use it. Jordan, your present didn't get logged in time. Okay. All right. So let's do some iClicker. All right. So we did, I think we were on, it's so hard to see with this thing on. We did that, we did that. Yep, it was 30, here we go, this one. All right, so here, this is our first one. All right, let's see if I can move this. And I'm gonna move this because whatever I see on my screen is what you see. I don't want this black thing to be here though. There we go. But this is still in the way. Go away. Well, I guess y'all can read it. Okay, so here we go. So we have a base pair. We talked about our base pairs last time. Which of the following nucleotides are present based on what you can see? And your nucleotides and your answer choices are represented by the single letters. And I tried to make it easy. A is A, B is G, C is C, and D is T. I'm gonna go close that door. app says I have 2 absences. Okay, I'm gonna look into iClicker attendance. We will see. How do you choose more than one? You can't. Oh, I do need to go and clean up. You're right, Ashley. You're right, Ashley. I need to go and clean up. You can only choose one. You only have to choose one correct answer. One correct answer. I think we had 55 people. 53? Okay, we'll call it here. All right, so you could have answered with either one, B or C. How did you know it's either B or C? Tell me in the chat, how did you know it was B or C? So it could have been B or it could have been C. Oh, I gotta stop it. Could have been B, could have been C. That is correct. Oop, go back. Okay, because we have 3 bonds. We have 3, what kind of bonds are they? Hydrogen bonds. They're not really bonds. That drives me crazy. It's not a real bond, but that's okay. So you have 3 bonds, and we know that our GC pairs have 3 bonds. How many do our AT pairs have? Only 2. Okay, good, good, good. So you don't actually have to memorize them, but which ones are purines, which ones are pyrimidines? Purine is the 2-ring, right? Pyrimidine is the 1 ring. Okay. Other 1. Okay. So let's try another one. Let's try another one. Okay. To break the base pair, blank energy needs to be invested that is required to break a base pair that consists of 2 hydrogen bonds. So if you're breaking a GC base pair bonds, does it require more or less energy than an AT bond? Okay, so I think we had 55. I think 55 is our number, okay? So should have been more. It should have been more. So good. Why is it more? Because we have 3 bonds to break, 3 energies to overcome. So it's going to take more energy to overcome 3 hydrogen bonds than to overcome 2. Wow. Dang it. I scroll on this one. Some of them you have to click, some you can scroll. So everybody just saw the answer to that one. You didn't? All right, well, let's see. Let's see. So we have an organism that's evolved to grow at significantly higher temperatures than most. What are you more likely to have? More uracil content in our genomic DNA, a greater AT content compared to GC, roughly equivalent AT content as compared to GC, right? Or D, greater GC as compared to AT. Okay, looks like everybody's in. So we're gonna stop it. And you should have said D. You absolutely should have said D. So why D? Well, if you're at higher temperatures, right, you want stronger bonds between your bases. So the more bonds you have, the more you can withstand high energy, and heat is energy. So you want to have more GC rich. Okay. All right. Next one. Let's see if I can do this. I keep forgetting that this one you can only, so click 1 correct answer. Click at least 1 correct answer. When within the double helices, right, that's what DNA is. Which relationships between both strands of DNA double helix are present? Choose 1 or more. But you can only choose 1, so choose a correct answer. Okay, so it looks like we got almost everybody. All right. So what you could have said is you could have said B. Oh, I didn't stop it. You could have said B or you could have said E. That is correct. All right. So complementary means on one side we have a T and then on the other side we have an A. It's not the same. It's not identical. It is complementary. So it's always A's and T's and C's and G's, right? Apples in the tree, cars in the garage. And it is anti-parallel. What are we talking about with anti-parallel? We're talking about the phosphate backbone here, right? And which carbon the phosphate comes off of versus which carbon the OH comes off of, okay? All right, which scientist generated an X-ray diffraction image of DNA extracted from calf thymus DNA? The image was called Photo 51 and helped to reveal the structure of DNA. And remember, if your clicker's not working, just log in the chat. Just put your answer in the chat. I know I'm not looking at it, but I don't want to show everybody your answers. Okay. So what do we got? Good. Oh, I'm so proud of y'all. You know who actually generated the image. The person who actually generated the image was Rosalind Franklin. Even though whenever we hear who discovered the structure of DNA, all we hear about is Watson and Crick, Watson and Crick. They didn't generate the data. Okay, you have to generate your own data, but you know. Okay, good. So let's go on. All right. See, I don't want to click this until that black thing goes away. There we go. In response to a change in our external chemical environment, organisms often experience a rapid alteration in gene expression patterns. Identify the terms that is or are not able to change rapidly within a changing external chemical environment. So what does not change quickly? Transcriptome, proteome, or genome? We haven't talked about those in class yet. The lecture, yes. But what? Oh, is the chat? Are these questions similar? Yeah, they're similar. So are the self-check questions. Okay. Doing good. All right, what do we got? 50 responses, a few more? I hate that that black bar comes up. I need to figure out how to not do that. All right, so I'm calling it. And what you should have said is that, ooh, go back, that the genome's not going to change rapidly. Is it going to change? Overall, is it ever going to change? Yes, absolutely it's going to change, but it's going to be a very slow change. If I could click the right things. Okay, it's going to be a very slow change, but the transcriptome and the proteome. So transcriptome is what you're making out of mRNA, right? The DNA to mRNA. And then the proteome is when you're going from the mRNA to the protein. So those things can change by the second, depending on what you had for lunch. So those have to be able to change very, very rapidly. But the genome, on the other hand, is a more slow, but more permanent change. Okay? Got a lot of conflicting answers in the chat. About what? They did my girl Rosalind so wrong. That's so true. They did. They really did. It was really sad. It was really sad. They need to award the Nobel Prize posthumously so that if you die, you still get it. Maybe you don't get the money associated with it, but you should get it. Okay, so what conflicting answers on what, Austin? Oh. No, it's okay. It's okay. You can get it wrong here. It's not a big deal. All right. Okay. So next one. I got to move this now. Okay. So consider our two images of primate phylogenies. Y'all have seen phylogenies before, right, in general biology. In terms of evolution of the species, blank phylogeny is more powerful at measuring genetic relatedness. We have morphological and molecular. More powerful at measuring genetic relatedness. I guess I can get rid of that. I don't need that at all. Okay. So what do we got? Oh, okay. So it's molecular. It's absolutely molecular. So if you look at the DNA, right, we're comparing human to chimp to gorilla. When you look at the DNA between a human and a chimp, it is so similar. There's way more sequence homology between a human and a chimp than there is between a chimp and a gorilla. So we can tell evolutionarily where we have branch points in the phylogenies just based on DNA. But if you look at the external features of a gorilla and a chimp are much more similar than the external features of a chimp and a human. But you can't just look at what's on the outside of an organism. You have to actually look at the DNA. This is what's really controlling all of the outward appearance and is how we define evolution. So you really have to look at the DNA if you want to know who's related more closely than others, right? Okay. All right. Y'all are funny. All right. So let's go on. I got a few left, not many. All right. Let's do this. Here we go. Upon reception of a biochemical signal by a eukaryotic cell, which of the following would change first when the genetic expression pattern of the cell changes? So what would change first? The proteome in A, B is the transcriptome, C, the proteome and the transcriptome simultaneously, or D, the proteome and the transcriptome would not change. Okay, getting close. Give y'all about 5 more seconds. Oh, there's everybody. Okay, I'm gonna stop. So what do we say? All right, so it is the transcriptome that is going to change first. So if you remember the central dogma, right? Everybody remember the central dogma? We go from DNA to RNA, from RNA to protein. So the transcriptome, we go from DNA to RNA first, then second would be the proteome, right? Okay. In terms of energy conservation by the cell, direct regulation of what synthesis is more efficient? Trying to check my chat. So a lot of these are really good questions where they're not necessarily asking you content questions, but they're asking you to interpret the content and apply it, which is what you'll see on exams. Okay, that looks like everybody. So what do we say? Ooh, it is the transcriptome. Why? Why is it more efficient to change your transcriptome than to change your proteome? Because it's first in the pathway. It's absolutely first. Y'all should be logging in, right? Y'all should be answering on here. It's first in the pathway. That's exactly right. the more energy you conserve, the earlier you change your pathway. Because it's really a waste to go to a proteome. You don't want to stop at the proteome because then you're making mRNA that you don't need. So energy-wise, we want to go to the beginning of our pathway, of our cycle. Oh, I messed that one up, right? What type of RNA delivers amino acids to the ribosome? It is tRNA. That was a memory one. All right, well, oh well. Okay, oh, this is a good one. Which of the following three terms includes the other two? Which one includes the other two? Homologous, orthologous, and oh my gosh. They're paralogs. Paralog. See, I just can't say it. I struggle with that sometimes. But homologs, orthologs, and paralogs. Almost there. All right. Okay, so what do we say? Oh, good. All right. So that is correct. A is correct. Homologs. If you have things that are homologs, the word homologous just tells you it's similar. It doesn't tell you why it's similar. It just says it's similar. If you're saying that two genes are orthologs of each other, then that's telling you that not only are they similar, but they're also in two different organisms. And finally, paralogs, we're saying that we're in the same organism and there was a gene duplication event. And that event resulted in two genes that look very, very similar. So all three are, oh, Emily, you want me to go back? I can go back, right? tRNA delivers amino acids to the ribosome. Amino acids, oh, and Caitlin answered you. Okay, good. Y'all got each other's back. That's awesome. I love it. I love it. Okay, so they're all homologs. All right. Okay, so that is it for the clicker. So what I want us to do, I'm going to escape out of this, maybe. I don't think so. I think because I'm posting your self-checks, I think I'm not going to post these. How do I get out of this? Okay, there we go. All right, so the next thing I want us to do is I want you to go to Moodle, and I want you to go to discussion part 2. Discussion part 2. Okay. Okay, I put us 2 rounds in case we get to 2 rounds, but I don't think we're going to get to 2 rounds. We'll see. All right. So what I did was I put together, I put you in groups, right? Here's hydrogen's group, and hydrogen has a question. Each of the groups has a question, and then I put each of you a space to kind of put some information, and then you need to put everything together and get to a final answer. So all of these are applications of what we covered in the lecture. So it goes through all of the sections, and then, and then if we have time, we have a second round, and these questions are a little bit deeper in thought. And so what I did was I put the groups together. So you actually have 8 people working on these bigger type questions. So, and the last one is one I don't even know that we can do in an Excel spreadsheet. It's to write out chemical equations. And so I'm giving brownie points. Do y'all know about brownie points? Brownie points are not real points. Brownie points are, they just make your professor happy. And then maybe she makes brownies one day when we're not in COVID. I used to do that for my other classes, right? You open the file and it's blank. You have to click on, there are little tabs at the bottom. Right, there's one that says groups, there's one that says round 1, and there's one that says round 2. So I want y'all to start on round 1. Oh, I did not mean to do that. Okay, Ross, did you get it? Now these questions you're gonna have, and you're gonna have the answers to. So I think the iClicker questions are the only ones y'all aren't going to have the ones to take home. So if you finish your round 1 and you have time, go on to round 2. All right, Ross, good to hear. Be careful helium, whoever's in the helium group. What types of molecules? Enzymes or catalysts, that's a function, not a type of molecule. Sodium, be careful and explain. You're missing some pieces in the middle. Yeah, we got a few people starting. Okay. Do we now have anybody in the sulfur group? So,
I'm working on the sulfur group, but it's really slow for me. I'm sorry.
are they working on this one? Okay, we do have somebody in there. Okay, good. Ooh, yeah, we're doing good. Be careful not to write over. Oh no. Yeah, you have to kind of look at what's highlighted and not type in a highlighted box. That's a good point. I'm sorry I didn't say that. Thank you, Adela. It's being really slow because there are so many people and it's not letting me type. Okay, if you want to go, if you're having trouble with that one, go to the round 2 and go and find yours for round 2 and then go back to round 1 and check and see if there's something you think needs to be added to the answer. And if you finish with all of your groups, go and look at some other ones and see if there's anything you think needs to be added. And if you want to add to somebody else's, don't type over theirs. Come over in this column H, and you can add some additional information here. If you want to give them, hey, I think you forgot this or that, go ahead and put it to the end. There are more columns. You can use those extra columns. That is no longer allowing you to edit. Can you exit the thing, close it, and go through Moodle again? It may freeze with the message trying to reconnect. I think the slides were better. You have to tell me what you think. Was it easier to work in slides or is it easier to work in sheets? Oh, slides. Okay. Overwhelming. Slides. The sheets are not letting me type. Okay. So we're just trying new things out, and I can stick to slides. Slides is fine. No problem. Got it. I listen to y'all. Y'all tell me what works and what doesn't work, and I promise I will listen and I will make changes. So slides was easier. Don't know until you try. People are deleting stuff without knowing. Okay, so slides next time. It happens. Okay. If you want to pick a square farther out, you know, you want to pick something out here and type it, just so that you know that they don't type over you. Oh, Colin. Oh, no. No. Okay, slides. I swear, next time, slides. Slides. Or I could put them, like, each group on a tab or something like that. That might work better. If we need something that's kind of an Excel-ish thing. So, but if we can do it on slides, we'll just do it on slides then. Austin, such positivity. I love it. Don't give in. Like the amount of people. But the slides were the same amount of people. And did y'all have that problem with slides?
Since everyone was on a different slide,
It was better.
yeah, then as of like one document.
And I bet it would be different if I had done like a tab for every group.
Yeah, probably.
But I think it's going to be easier. But I think it's going to be easier for y'all to. They're not all on one page. Yep. That's exactly what we were saying. And I think it's going to be easier to collect the information for y'all to keep on the slides. Like it's easier to look at later than having 20 different tabs at the bottom of a sheet. So I think slides was way more successful. So we're going to try slides. All right. So we got 1 minute left in class. So what I want you to do is I want you to finish this, because this weekend I'm going to go in and I am going to look over all of y'all's work and put your comments and stuff like that. So this is a live working document that will stay up and will stay live. So finish up what you didn't finish, and I will check over them this weekend and then be waiting. I'm going to send you an email that has the next set of lecture videos. We're on chapter 2 next. All right, y'all have a wonderful weekend. It was great to sort of see you, and see you on Monday. How would you study from something like this? This gives you examples of how questions can be structured such that they're not going to be complete recall questions. They're going to be way more application. So you might have some where you have to recognize, okay, what's this base pair or something like that. But more of them are going to be, why is carbon so important in the formation of our organisms, right? We've talked about all that in class. So it's more about application. So understanding the material, and then this is practice applying it. Okay, you have a great rest of your day too. Happy Friday. I did have a question. Okay, what was your question? A question about one of the questions.
Can you hear me? Dr. Bergeron?
You can go ahead and ask your question. I don't know if you're typing. I can't see when y'all are typing. It's hard for. I think she has us muted. Oh, do I have y'all muted? Hold on. Hold on. Because I could put y'all on. It's not security. I do have y'all muted. Let's see. I don't think your computer can hear it. Oh, maybe it's the volume here. Okay, try again.
Can you hear me now?
All I hear is background noise. I don't hear y'all. PC. The volume's up. I mean, oh wait, I heard somebody.
Hey.
Hey.
Okay.
Hannah?
Yeah, that's good.
Yes, this is me.
Okay, so I hear Hannah. All right.
All right. I don't have any questions, but I want to make sure y'all had it. So y'all take care.
Thank you.
No problem.
Okay, so who else has a question? You can try to unmute yourself and let's see.
Can you hear me now?
Oh, it works. Yay.
Okay. So earlier, one of the questions, it was asking about, it was like the 2 base pairs, and it said like the greater one would be the G and C, but I don't understand why it would be that one.
Okay, let me pull it up. I know which one you're talking about. Okay. This one? Well, this one really, right?
No, keep going. It's not that one. It was like it was 4 different, it's 40.
40, slide 40?
Yes.
This
I
one?
don't understand why it's D.
Okay, so if they're at a significantly higher temperature, right? The thing with temperature is the higher the temperature, the easier it is to break bonds. The more energy you put in, the more bonds you're going to break. and an AT pair only has 2 bonds. So we call it melting DNA. Have you heard of that before?
Mm-mm.
So if you melt DNA, you separate the strands from each other.
Okay.
So at a relatively low temperature, AT base pairs will melt, will separate.
Okay.
But it takes a higher temperature to melt a GC because you have 3 bonds instead of 2.
Okay.
So an organism that has been growing in a high temperature environment for a long time is going to evolve to have more GC base pairs than AT base pairs because they have a higher survival rate.
Oh, okay, okay.
It's
That makes sense.
it's better to live in that environment with 3 bonds than 2. Now, their whole DNA can't be GC, or they wouldn't be able to make proteins. They wouldn't be able to do things. So you have to have a mix. But if you looked at like our percentage of GC versus the GC content of organisms at a geothermal vent, trust me, our GC content is much lower than theirs. They have a much higher GC content. So they would have D, the ones at the geothermal vent.
Okay.
Good?
Thank you. Yes, ma'am.
Yeah, no problem.
I also have a question.
Sure.
It was about the 2 before it.
So wait, let me turn you up because it's a little bit low. Okay, go ahead.
The 2 questions before this one. The
2
first question I
questions
had.
Okay, I'm gonna go up. This one?
Yeah.
Okay.
So for this, I didn't really understand the question and like the way it was being asked. Like, I don't know if I'm missing something, but like, I didn't really get what it was like.
So what it's asking you is, okay, here's a picture of 2 bases, right? Now, how do you know what one of these 2 bases is? We know, you have to, number one, know that A and T base pair together and G and C base pair together, right? Those are the only 2 pairings that we have.
Yeah, I understand that part.
Okay, so
but I guess I don't really get how to translate that to looking at the molecule.
okay,
You know?
so there are really only 2 pictures that I could put up here. I only have one. I can pull up the book. You know what? Wait, let me pull up my lecture slides, which, oh, I don't know if I have on here. Oh, I do. Yeah, no, that's 109. Oh, crap. Sorry. That's my wrong class. I need to go to 435. I don't know if I put our lecture slides, but I'm gonna answer that. I need to show you another picture to answer that. Let's see. Oh, but I think I put y'all the slides, the lecture notes. Here we go. Okay. I put them for you. Okay, so if you go, keep going, keep going. Where was that? Here. Okay, this one right here. So these are really the only 2 pairings that you're ever going to see, because you don't know which strand the guanine's on and which strand the cytosine's on. It doesn't matter. It could be a mirror image, but it's still going to be the same pairing, right?
Yeah.
Okay, and the same thing's true about an adenine and thymine. It'll either be this way or it'll be flip-flopped with thymine on one side, on the other side, and adenine on the other side, right?
Okay.
Okay, so when you look at these, you can tell that this must be a GC pair because you have
It has
more,
the 3, yeah,
and adenine and thymine are always going to have
2.
2. So
Yeah.
that's how you can tell. That's why it's easier to answer your question by looking at the other structure.
Yeah, okay. And then that was also why the second question was about which one takes more energy to break.
Correct. So this
Okay.
one takes more energy to break because you're overcoming
3 bonds.
and they're not real bonds. You're not really breaking anything. You're really overcoming intermolecular forces. And I really wish the whole of chemistry would change the way they call that, but that's never going to happen. And that's just one of my pet peeves. But it's overcoming the hydrogen bonding intermolecular force. Yes.
Okay.
So it takes more energy to do it here, less energy to do it here.
Yeah. And then that also has to do with like the temperature that she was explaining to the other girl earlier.
Well, what is temperature?
Okay, yeah, you're right. It's like
Temperature is energy.
yeah, the energy. So the more energy you use, the higher the temperature is gonna be.
Correct.
So yes, that makes a lot of sense.
Yeah, but you got to get that first one before you can get
The rest of them, yeah, okay. All right, thank you.
Of course, of course. Any other questions? Let me look in the chat. Okay, if we are meeting online. Everyone, if you're taking my 437 biochem lab, everyone is meeting in the lab in the room next to the lab, which is 210. But just come to the lab area and I'll sort you. But everyone come to lab. Okay, any other questions? I'm going to close the Zoom if there are no other questions. All right, Logan, so I'll see you this afternoon. Go get some lunch.
Can you hear me, Dr. Pazra?
Yes, ma'am.
Do we have to come up with a final answer for our group on Google Sheets?
Do you have to come up with,
The final answer in the sheets.
yeah, I was hoping that y'all would, but just kind of by looking at everybody's answer. So you should be able to maybe even copy and paste one of them, you know, or most of one of them and maybe add something else. That's kind of the goal. I'm going to look at them and over the weekend. And so I'll comment and y'all can continue working on them. I mean, this is not something that I'm going to take away from y'all. You're always going to have it.
Okay, so should I put in the final answer for my group?
Sure. If you want to copy and paste whatever you think is the best one and dump it in, that'd be great.
Okay, thank you.
Yeah. Okay, we have another question. Yes.
For the final answer, if you have 4 parts for one question, how do you sum all that up just to a final answer? Just copy like a little from each one. I'm in group 4, beryllium.
Okay. You're in beryllium. Okay. So, yeah, everybody kind of has some interesting, yeah, you kind of have to.
It's the I think group 6 or so, I'm not sure.
Yeah, and I'm not going to take away the individual. You were group 6? Oh, beryllium.
Yeah.
I see.
7.
What are the 4 major types and briefly describe them? Yeah, so some of the answers are going to be bigger than others, right? But
Right.
yeah, and if you wanted to have each one in one of these and then in your final answer say, you know, each biomolecule is in one column or something like that, that's fine. I mean, I want this to be something helpful to you. That's the whole goal.
Yeah, got it.
So whatever works best for y'all, I'm good with. I'm really flexible on that.
Okay. Thank you.
Yeah, no problem. Okay, let's see. Anybody else? Okay. And I didn't look at the chat just to see if anybody else put anything in because I can't make it stay up. All right. So we're good. Okay. So I will see y'all on Monday and some of y'all in an hour. Okay. I'm going to end.